So hello friends, today I am going to teach about the CRISPR-Cas9 system. So this is a very good technique to achieve the desired gene insertion in a particular location inside the gene, inside the DNA of an organism. Because other techniques are there also, but CRISPR-Cas9 एक ऐसा टेक्निक है जो एक पर्टिकुलर साइट पे आपको एकदम मीन्स कहते हैं ना एक्सप्रेशन ऑफ जीन एकदम अच्छे से देता है। ये बहुत स्पेसिफिक होता है। इट विल ओनली इंसर्ट ए पार्टिकुलर gene inside the DNA at a particular location. So that's the beauty of the CRISPR-Cas9 as compared to other gene delivery methods like electroporations, uh, transformations. There are not more transformation. Electroporation is there, and the agrobacterium mediated gene transfer method is there. So there are a lot of off target effects, right? So in CRISPR यहाँ पे एक एकदम स्पेसिफिक जीन आप ट्रांसफर कर सकते हैं पार्टिकुलर लोकेशन पे एंड डिजायर्ड आउटपुट ले सकते हैं। आप नॉकआउट कर बना सकते हैं, आप नॉकडाउन बना सकते हैं। सो नॉकआउट और नॉकडाउन हम दूसरे वीडियो में डिस्कस करेंगे। अभी हमारा मेन टॉपिक डिस्कस करने का है वो क्रिस्पर है। So basically CRISPR is an advanced tool for editing editing in the plant genome or human genome, right? So CRISPR में क्या हम देखते हैं? CRISPR genome editing एक site specific nuclease है। It's a type of genetic engineering in which DNA is inserted and replaced or removed from a genome by artificial engineered nucleases, right? So this is the definition of the CRISPR. So there are various kind of gene delivery systems. The first one is non-biological, second is biological system. And non-biological systems are basically chemical methods like lipid-mediated transfection, where we use some transfection agents. There are a lot of commercially available transfection agents in the market, and there are calcium phosphate transfection. And there is physical method like electroporation, where we use electric current voltage, where we just create pores inside the bacterial membrane to transfer the gene inside the bacteria, right? And we can also do it for cells. So this is electroporation. Electroporation is a bit harsh for the cells because cell comes under stress and dies sometimes, right? You will get a lot of cell death. So this is non-biological based. Another one is the biological based systems like lentiviral systems, where you just clone your genes and express your proteins and inside the cells, right? Because it is lentiviral based systems, so we get proteins. And there is adenovirus based systems, and adeno associated virus based systems are also there, right? Then you see comparison between the prokaryotic and eukaryotic systems. So basically in prokaryotic system there is a tracer RNA, and there is CRISPR cluster regulatory interspace palindromics repeats, right? So it is transcription, and there is formation of the pre-CRISPR CRISPR RNA, and there is tracer RNA formation. Then CRISPR RNA processing is there. Then unidentified nucleases is there, which is not identified yet. Then there will be DNA degradation. There is a target DNA, there is a CRISPR-RNA, there is Cas9, and there is a tracer RNA. As you can see in the figure, they are chewing out the viral bacteria. Basically, they have shown the bacteriophage, right? Bacteriophage DNA. And in another eukaryotic system, there is engineered one CRISPR-Cas9 cassette is there, which is basically composed of Cas9 and guide RNA. Guide RNA you have to design according to your wish. Suppose you want to knock out a gene X, you have to design the guide RNA according to that gene, so that it guide RNA guides it towards the particular location, and it just that location is cut and your gene will be inserted in that position. There are a lot of available commercial vectors inside the market. In the market, you can just purchase and design your experiment. It's not that difficult. This is just to follow some literature. So there is a guide RNA in the eukaryotic system, and there you will get the guide RNA. There is a transcription, and that again the transcription will convert that the guide RNA will guide the Cas9 to the particular location, and Cas9 will cut that particular location, and it will just silence the gene right at the target loci. It depends upon what you need to do. Whether you need to knock out or knock down the cells. If you knock out, then that gene will be totally silenced. If you knock down, that the gene's expression will be low. So this is the basic difference between the knockdown and knockout cells, right? In the cells. So there is cells endogenous repair mechanisms for double stranded breaks like non-homologous end joining like an HEJ and homologous directed repair like HDR. So in an HEJ there is no donor DNA is there, and there is a donor DNA. If donor DNA is present, there is it is cut in the middle and that is inserted inside the gene. If donor DNA is there, then there is gene knockout. In case of the homologous recombination, there is donor DNA and there is uh, one, uh, one other DNA and there is genetic modifications. And in case of the plus donor DNA, there is also genetic recombination and there is gene insertion. So these are two recombinations like NHEJ and homologous recombinations. So these are the sequence of events during the CRISPR-Cas9 system. First one is PAM is recognized. Then this is guide RNA guides it, and basically it cleaves. It is cleaved by your Cas9 system, and there is at that where your Cas9 is guided by the guide RNA and it replaces your gene of interest. There will be total silencing after that part. Just like you switch off a bulb, you will switch off the bulb and bulb will off. Just like the whole function of the gene downstream to that gene will be totally switched off, right? So CRISPR-Cas9 transporter of the enzyme to specific DNA sequence. So dead Cas9 transporter other enzymes attached to it in a specific DNA sequence. So there is active Cas9 and there is single Cas9 domain Cas9, as you can see in the figure. There is dead Cas9. So it is not used, right? So CRISPR-Cas9 is beyond genomic editing. So it basically helps in the regulation of the gene expression. Dead Cas9 can be recruited to the specific DNA sequences such as fusion protein with transactivation or trans repression domain or transcription factor. Then there is visualize selected loci living cells. Dead Cas9 can be fused with the fluorescence protein to visualize 3D architecture of the genome, right? Now, advantages of the CRISPR-Cas9 system. This is simple, accessible and more versatile tool than the zinc flinger nucleases and talents, right? So it basically creates variation of a desired loci. You will get a very precise result. CRISPR-Cas9 also allows multiplex gene editing by simultaneous expression of 2 or more guide RNAs, and it allows the inexpensive assembly of large guide RNA so that CRISPR-Cas9 applications bring in genome editing within the budget of molecular biology laboratory. The CRISPR-Cas9 system can introduce biallelic or homozygous mutations directly into the first generation, and being easy and affordable tool, CRISPR-Cas9 promises to revolutionize basic and applied plant research. So these are some of the advantages of the CRISPR-Cas9 system. Now, what are the drawbacks of the CRISPR-Cas9 system? We have talked about a lot of advantages of the CRISPR-Cas9 system. One of the few criticisms of the CRISPR-Cas9 technology is relatively high frequency of the off-target mutations also, reported in some of the earlier studies, as it is very precise in nature. But sometimes it is reported that there are a lot of off-target mutations also. The limited data available thus far suggests that off-target effects are rare in plants. These are off-target effects are rare in plants which can be eliminated by backcrossing again and again. that is progeny is crossed again with the parents, right? Then off-target effects vary even among different cell types in the same species. Some people don't understand what is off-target effects. So it's easy to understand actually. You design a gene for particular, design a guide RNA for particular gene knockdown, right? And that guide RNA will knock down lot of different genes inside an organism. So that is called the off-target effect. Okay. So what are the strategies to reduce the off-target effects during genome editing? So these are some of the, this is very important actually, because when you design an experiment, you should be practical, because if there are a lot of off-target effects, you will not get desired results and you will just roam in experiments and you will waste your time. So mutated version of Cas9 has been produced with D10A mutation in RUVC nucleus domain that convert into any nicases, right? The use of two Cas9 nicases can therefore generate off single strand nicks that produces staggered double strand break. Potential off-target sites were unlikely to be sufficiently close to each other to induce the more than individual nicks. So this is the way to, as you can see in the figure. So this is the way to reduce the off-target effects. Now another point is that altering the length of the guide RNA can also minimize the target modifications. So guide RNA with two additional guanidine residue at the 5' end, as you can see in the figure, and we are able to avoid off-target side effects efficiently than normal guide RNA. So you have to add GG in the 5' end of the guide RNA, but we are also slightly less active at one target sites, right? So this you should also keep in mind. Now what are the applications in plant breeding? So accelerated plant breeding. So you can achieve the accelerated plant breeding by allowing the introduction of precise and predictable modification directly into the allied backgrounds. So you can create multiple traits can be modified simultaneously, and non-homologous end joining mediated gene knockouts are the simplest form of targeted modification you can do. And the insertion of the large sequence of non-homologous end joining or homologous recombination would allow the introduction of transgene at loci that promote high level of transcription and do not interfere with activity of endogenous genes. And this also allows targeted modification trait stacking. Plants altered by the excision or insertion of few nucleotides using CRISPR-Cas9 would be classified as genetically modified organisms. So there are target insertion of transgenic into the fields of metabolic engineering and molecular farming. Okay. Now these are the some of the applications of the CRISPR-Cas9 in plant. I am not going to discuss all of that. You can just take a screenshot and you can see later. You can search papers and you can just browse through the techniques, and you can just start planning your CRISPR experiments. Okay. So these are the various case studies which are done on CRISPR. Like first one is in by Nature Reports, rapid breeding of the parthenocarpic tomatoes using the CRISPR-Cas9. You can read the paper. It's a very good paper. So here the objective was to introduce parthenocarpic tomato by distribution of the S2AA9 gene with CRISPR-Cas9 system. Tomato varieties were used like microtom. Design of target guide RNA. In total, the 5 guide RNA and 3 sites were selected for the CRISPR-Cas9. So these are the CRISPR-Cas9 techniques, and the detection of the CRISPR-Cas9 induced mutations are done. Okay, you can just read the paper. So these are the CRISPR-Cas9 knockout phenotypes they have selected. Now what is the conclusion? So the simplicity and accessibility of CRISPR technology platforms provide many advantages, and it is very specific in nature, though there are a lot of off-target effects which need to be minimized every time you plan your experiments. And together with the other sequence-specific nucleases, CRISPR-Cas9 is a game-changing technology that is poised to revolutionize basic research in plant breeding. CRISPR-Cas9 has already changed the face of research, but what's been achieved so far could just be the tip of iceberg. when it comes to CRISPR potential, right?